Hey, hello, good evening after this beautiful spring/summer day at this point. I'm Amy Rowe, and I am a county agent with Rutgers Cooperative Extension. I'll be your host for this evening of our Earth Day Everyday and Marine Extension Program Seminar Series collaboration. So welcome to our May uh, version of this presentation with the land talk happening today, and next week we will have our marine talk for May. But yeah, I wanted to welcome everyone to Earth Day Every Day, and we will be hearing from our speaker in just a moment, but I want to go over some logistical things. So you may notice that the chat is disabled. We would like you to put questions in the Q&A section if you have any questions, and we will get to the questions about halfway through the first half of our presenter's presentation, as well as at the end. So don't worry, we will try to get to as many questions as possible, but just wanted you to know that chat is disabled. This is just Q&A, and we will attempt to get to as many questions as possible. So we are very excited to be focusing on all the small things with big environmental impacts. Again, this is the land version of this talk today, of this presentation today. Go ahead, Donna, on the next slide, please. So we will be administering a poll towards the end of this presentation in order to understand what you have learned from this program, as well as what actions you will be thinking about taking after hearing this presentation. And in order to administer that poll, we need your consent to take part in this research study. We understand that you are human subjects, and we very much care about protecting you. So we are following the International Review Board protocol for protecting those human subjects. So we would like you to consent to take part in our study. If you have any questions, you can email or contact me, Amy Rowe, down at the bottom there. But we would like you to stick around and take the poll so that we can make this series better and we can learn what you have been learning. So please take the time to do that. We will be popping that up towards the end of this talk, so please participate if you feel comfortable doing so. Go ahead, Donna. Also, Rutgers Cooperative Extension is an equal opportunity program provider. We are in accordance with federal law, as well as the U.S. Department of Agriculture's civil rights regulations and policies. So we do not discriminate on any of these things based on race, color, national origin, all of those things. So please, if you have a complaint, here is where you can complain. But our programs are open to everyone, and we're very happy to have you all here joining us tonight. So just be aware of our equal opportunities and that our programs are open to everyone. Go ahead, Donna. So now it is my pleasure to introduce Dr. Donna Fennell. She is our speaker for tonight. She is the chair of the Department of Environmental Sciences at Rutgers University. She is housed in New Brunswick. She has been a professor. She is a full professor now, and she has been doing all kinds of things related to environmental engineering and environmental sciences, and her main focus of research is in microbial processes in both engineered and natural systems. So please welcome Dr. Donna Fennell, and we will get right going with the presentation.
Okay, thank you so much, Amy. It's a real pleasure to be here. I really appreciate the invitation. So yeah, I'm going to talk to you about bioremediation, just a little bit about our department. Environmental Sciences at Rutgers is the oldest environmental science department in North America, and we celebrated our centennial in 2020. Our students participate in programs in environmental science, environmental engineering, and atmospheric science and meteorology. So that's just a little bit about us. But I'm going to go right into talking about bioremediation, one of my favorite things. So what is bioremediation? It's a technology and often an engineered technology that modifies environmental conditions to encourage microorganisms to destroy or detoxify organic and inorganic contaminants. So we modify those conditions by trying to change the environment for the organism to enhance their activity. And I'll show you a little bit about that. So first of all, what are microorganisms? So probably you all know this already, but microbes are considered organisms of a microscopic size. They may exist as single cells or as a colony of cells. And you see there on the left, a tree of life that was made by Carl Woese in 1990 when he began to use the sequences of the nucleic acids in living organisms to classify them into different groups. So this resulted in the three domains of life that you see there: the bacteria, the archaea. Both the bacteria and archaea are microorganisms. They're generally speaking single-celled, sometimes growing in chains or colonies. And then we're over there in the animals, in the eukaryotes. So the microbes are a very important part of our biosphere, and bioremediation generally harnesses mainly the bacteria for degrading pollutants. Sometimes fungi can be very useful, and the archaea tend to be more just players in the broader community, but they're always there in those environments as well. So one of the things about bioremediation, where we're trying to use microorganisms to degrade pollutants, is that sometimes it's done in situ, meaning we leave the contaminated soil, groundwater, whatever it is in its place, and we try to treat it where it is located. Sometimes we do this treatment ex situ, where we remove the contaminated material from the original location, we take it somewhere else, and we do that stimulation to get microbes to grow and enhance bioremediation and degradation of the pollutants. So when we think about bioremediation, there are a number of different approaches where bioremediation is important, and those are called natural recovery, biostimulation, and bioaugmentation. But for any given contaminated site, we could use a combination of these approaches. So let's look at natural recovery. So natural recovery is sort of what it sounds like. Let nature take its course. We know that there are microbes that are naturally occurring. They're everywhere. And when pollutants are spilled, they will begin that degradation process. So sometimes those natural processes that might also include things like just dilution of the pollution, or having the pollution move from water into air and thereby be diluted. There could be some chemical processes that result in the transformation of the pollutants. But microbial processes can also be a part of this natural recovery. Sometimes that's enough to keep pollutants from harming us, to keep them out of someone's drinking water, or keep them away from important fisheries or agricultural areas. But sometimes natural recovery isn't enough. Sometimes we need to intervene in that process. And when we do that, it's called biostimulation. So during biostimulation, we try to listen to the microbes, so to speak, find out what it is they need to begin to degrade pollutants. And then we add those things to the contaminated water to help the microbes live and grow and be able to degrade the pollutants. So the engineered aspect of bioremediation then often is trying to figure out, how do we add the right stimulants to a contaminated groundwater, contaminated soil, contaminated sediment that will reach the microbes, reach the pollutants, and enhance that biotransformation and bioremediation. So let's look at what biostimulation might look like. So if you look at a person, right, we eat organic matter like a donut. I know I shouldn't eat a donut, but I love them. So let's say I'm going to eat a donut. That's my energy source. That donut is pure organic matter, just about. It gets oxidized in my body to CO2. That oxidation generates energy. I might grow new cells or make bigger cells, right? And coupled to that donut, I need to breathe in oxygen. Oxygen is my electron acceptor. It's what's coupled to the donut to make energy for me to live and be able to function. So we are very limited, right? We can eat organic matter and we breathe oxygen. That's how we make our living. And that's the only thing we really do. But the bacteria are so incredibly useful to us because they do many other things besides just eat organic matter and breathe oxygen. The bacteria make up the second largest mass of living organisms on Earth. They're second only to the plants. And while, as I said, we live on oxygen and donuts, or broccoli, if you're living in a more healthy way, the bacteria can live on many other food sources. They can breathe other compounds besides oxygen. So I'll show you a little bit about what that looks like. Okay, so here's a microbe. And let's say we're going to do stimulation, and that compound that's been spilled, the pollutant in the environment, is an organic compound. And examples of that are things like oil spills or gasoline spills. The many hundreds or thousands of different chemical compounds in those types of contamination events are organic in nature. And so the organism can oxidize those to CO2, generate energy, and grow new cells. But we need to stimulate them to do that. So one way to do that is to make sure they have enough oxygen. So we could provide some electron acceptor like oxygen to them so that they can live and grow. That's an aerobic environment when we provide that oxygen. Sometimes an environment is so contaminated that all the oxygen gets used up, and we shift to other electron acceptors. And the bacteria, some bacteria can transfer and start using other electron acceptors, or maybe they don't use oxygen at all and they prefer another electron acceptor. And examples might be things like iron. There are iron-reducing bacteria that will degrade compounds in fuel spills. Nitrate reducers or sulfate reducers can be operative. So bacteria are useful because they have the ability to shift and use other things in the environment. And so when we do biostimulation, we may be providing oxygen or nitrate or sulfate, something like that, to them to degrade organic matter. Now, there are other organisms that I'm going to talk about a little bit later that breathe chlorinated contaminants. So instead of oxygen or iron or nitrate, they're actually using a chlorinated toxic contaminant in the way that we use oxygen. They breathe the chlorinated contaminant. And so if we want to stimulate those types of organisms in the environment, if we've got a spill of a chlorinated compound that's a pollutant, then that means we need to provide them some organic matter that they can utilize as part of their energy making process. So in biostimulation, we're trying to figure out what is it that we want the organisms to degrade, what do they have, and what are they missing? And then we add the missing material, the missing compounds. Now, another approach to bioremediation is that we may add the microorganisms themselves to the contaminated environment. And there are microbes everywhere. There are organisms that transform many different types of pollutants. They're widely distributed. They may be present at almost any site. However, sometimes they're present at kind of low levels. And so to speed up bioremediation, we may add more of those microorganisms to a contaminated groundwater, contaminated sediment, contaminated soil. In addition, we would add those biostimulation agents, adding oxygen, adding an organic compound, whatever it takes for those particular microbes to be active. And there are commercial bioaugmentation agents. I always love the ads that are very clever about little microbes that you can buy on a commercial scale. They're grown in culture, they're containerized and shipped all over the world to be added to contaminated sites. And some of these organisms are really highly novel and highly specialized, and they do very specific things for us in the bioremediation process. So where is this contamination? Well, it's basically can be everywhere. We know that, right? We know there are contaminants in air, in soil, maybe in agricultural soils or urban soils. We have different kinds of contaminants. We've got contaminants in surface water, in the sediment, the aquatic sediment at the bottom of surface water bodies. And then a big one is groundwater. So the water that fills the spaces in the subsurface that we may use as drinking water through sinking wells and pumping that water out. Groundwater can also be very susceptible to contamination. And so bioremediation can be used in all of those different environmental compartments, not air yet, but interestingly, air is also full of microorganisms, some of which probably do degrade pollutants in the atmosphere. Okay, so I want to give you an example that we do in one of my classes that illustrates a type of organism that is very important for bioremediation. So we're going to look at how our students tracked what's called dechlorination. So dechlorinating bacteria, as I mentioned before, live by removing chlorines from toxic compounds. They breathe the chlorinated compound. They don't like oxygen. In fact, oxygen will kill them. And so they need an environment that's free of oxygen. They're naturally occurring, and they live wherever chlorinated compounds exist. And because there are many naturally occurring chlorinated compounds that are part of the natural biosphere, these organisms are widely distributed. So their name really rolls off the tongue. They're called organohalide respiring bacteria. So they respire organic compounds that contain halogens like chlorine. So what students did was they went out and tried to find these in Rutgers soils. So here's an example of a step-by-step dechlorination process. Okay, so that top compound that you're seeing there, it's got 2 carbons double bonded, and it's got 4 chlorines on it. And it's a toxic carcinogenic compound called tetrachloroethene. It's also called perchloroethene or PERC, and it's dry cleaning fluid. You're probably very familiar with how it smells if you've ever had wool clothing cleaned. So what dechlorinating bacteria do is, in a stepwise manner, they knock chlorines off. So they can go from the 4 chlorine compound, they remove a chlorine, and now they've made trichloroethene from tetrachloroethene. So trichloroethene itself is a very common groundwater pollutant. It's an industrial solvent, and it's also toxic and carcinogenic. Then they stepwise remove another chlorine. Now we've got dichloroethene, also used in industry. Another step, we've got a monochloroethene molecule. You might know that as vinyl chloride. It's a very highly carcinogenic compound. It's also very volatile, so it's generally a gas if it's allowed to evaporate. And it's used to make all sorts of plastics. We've heard about it a lot since that East Palestine train wreck. It's used to make polyvinyl chloride, a polymer of those vinyl chloride molecules. And I'm told it's responsible for new car smells. So if you get a new car and you love the way it smells, apparently that's vinyl chloride. And finally, the dechlorinating organisms. So far, all they've done is take toxic compounds and turn them into other toxic compounds. But that final step is where they really come through for us. Because if they knock off that final chlorine, they make ethene. And ethene is a gas that is harmless. It's made by ripening fruit. So it's always shown in these types of talks next to a banana. So we know that it's not harmful. So if we take any contaminated, if we take any environment and contaminate them with one of these chlorinated compounds, naturally occurring organisms will begin to knock those chlorines off in their respiratory process. And if we keep providing them with the organic matter that they need to couple to this, then we can get that process to go all the way to ethene, and we've detoxified the environment. So we do a lot of microcosm studies of these organisms. I hope my video is going to work here. So here's my student, Melissa Caruana. The first video there, she's making some special growth medium for these types of organisms. She's made the conditions go from aerobic to anoxic because they do not like oxygen. In the next video, she's there, hopefully the sound's not going to be too loud, but she's there setting up some larger scale microcosms. She's added some mud from, I believe in this case, the Hudson River to these bottles. She's adding the microbial growth medium to those bottles. And she's got gassing going on to keep the bottles oxygen free so we can grow the types of organisms that we wish to study. So any in microcosm that you make, you would take some of the mud, you would add a pollutant of interest, and then you would try to track those dechlorination products that we saw in the previous slide. So we set up lots and lots of these microcosms because we study different sites and different contaminants. And, the trick is really to try to keep the environment close to what the organisms might experience in the actual environment, although that's not always easy for us. That's what we try to do. So there you see just a little bit of the lab work that's being done in our research lab. So we had students from my various classes, either my environmental engineering senior process class, or I used to run a burn seminar. And we had students go out and collect soil samples from all over Rutgers campuses to see if we could detect dechlorinating bacteria in those soils. So we set up lots of microcosms and we monitor them in different ways so for example, seeing how much biogas a bottle might make. We might measure the volume of biogas. Here you see these anaerobic microcosms. As they degrade organic material, they make CO2. They may also make some methane. And so the bottles will become pressurized with gas. And that's one of the reasons we use these special type of serum bottles to study these organisms, because it can hold a lot of pressure. Now, in addition, we may also, use gas chromatography to obviously to measure the chlorinated compounds. So here's a map of some Rutgers locations. Down here, the green at the bottom on that map is the Cook campus where I am. You can see, where New Brunswick is and Highland Park. The blue is Livingston campus. The purple up there is the Bush campus. And students brought soil samples. You can see them in their little microcosm bottles from all over the place. And then we added trichloroethylene to each bottle. And we looked at that dechlorination of the trichloroethylene down to ethene. And you can see that the soil samples they brought, showed a variety of different activities, but it always does a little bit of dechlorination. There's not a single soil sample where nothing happened. So what that means is that these dechlorinating bacteria are just fairly ubiquitous. We find them in lots of places. Okay, so let's look at a more serious bioremediation example. We're going to talk about another chlorinated compound, dioxin. And dioxin is a major contaminant in the lower Passaic River. You've probably heard about this. So what are dioxins? Well, here's their chemical structure. They're a much more complex looking molecule. They have these two rings joined by oxygen bridges, and they can have up to 8 chlorines on them. So up at the top, you see that ClN, that N and M go up to 8. On the bottom, the compound that you're seeing is called 2,3,7,8 tetrachlorodioxin. Oftentimes, it's just referred to as dioxin. It's considered the most toxic and dangerous of the potential chlorination patterns that could be occurring with this class of compounds. So dioxins were never made on purpose. However, they're naturally occurring. They're part of the natural earth. They get formed during natural forest fires. They're formed in volcanoes. But as humans, we've done, an awful lot of done a lot of processing of chemicals. We've burned things. We've burned garbage. We have house fires. We have the train wreck that you've heard about recently, and so we cause a lot more production of these dioxin molecules than do natural processes. So through inadvertent production, they're quite ubiquitous, but there are across the world a few cases of very serious contamination by dioxins. So the reason that dioxins are, first of all, they're very toxic to humans, birds, mammals, and other mammals and fish. They cause cancers, birth defects, many different terrible effects that they have. And they're toxic at very low levels. What they do, because they're these big, heavy molecules, they like to stick to soil and sediment particles. So they tend to accumulate in the sediments at the bottom of bodies of water. And they like to partition into living organisms. So they like to, for example, go into fats and lipids in living organisms. So they bioaccumulate into small benthic organisms, which are eaten by maybe little fish, which are eaten by bigger fish, which are eaten by us. And they bioaccumulate and biomagnify up the food chain. So our main exposure to dioxins because of this is through the food we eat, especially things like fish. But if dioxins are deposited in soils, they may get incorporated into grains, which get fed to cattle. And so we may get some exposure through other types of food as well. So and there you can see a nice picture of some sediment. This is actually from the Anacostia River, not the Passaic River, but the Passaic River sediment looks very similar to what you see there in my hands. So why is the Passaic River and dioxin such a problem? Well, here's an overhead map of our New York, New Jersey harbor estuary. You may know all the names of the rivers, but let me put them on there. So, the big one up at the top that looks very muddy, that's the Hudson River. The Passaic River is the little smaller one. It's got that nice bend on it. We see the Raritan River over emptying into Raritan Bay. We see Staten Island and Brooklyn and Manhattan. New Jersey's down in the lower left corner. We're probably just off the map. But what you see in this image is also why sediment contamination by things like dioxin is so harmful. You can see the sediment plume coming out of the rivers, especially the Hudson, which seems very full of sediment at the moment when this image was taken. And that sediment gets transported down into the harbor. And of course, there's tidal action, right? So all of these coastal rivers are tidal. So the tide goes up, the tide comes out. And so this entire New York, New Jersey harbor complex, we're seeing mixtures of sediment. So the sediment from the Passaic that gets transported down, it might go a little way up the Hudson and vice versa. So any dioxin contamination in the Passaic is very harmful in the Passaic, but it also spreads around. And because those compounds are so toxic at such low levels, even a small amount of spread can be serious because of that effect that it has of bioaccumulating into fish, for example. All right, so in the Passaic River, the site of the dioxin contamination is called the Diamond Alkali Superfund site. It's basically in downtown Newark, right on the river. And here's sort of a picture of the Passaic River that I took on a kind of a gray day. But the river does tend to be, fairly full of sediment. It looks a little, dark, a little muddy. When you go by train to New York, you go over this river and you might notice that it's not as clear as the Raritan is on some days. And that tidal action is very easy to see. You can be standing at the river and see the water go upriver from the tide coming in. All right, so why is there dioxin in the Passaic River? Well, the Diamond Alkali Company produced what are called tactical rainbow herbicides. And the most famous of these is Agent Orange. So Agent Orange is a mixture of two herbicides, 2,4-D and 2,4,5-T. And during the manufacturing of those herbicides, dioxin, which I've shown here as that 2,3,7,8 tetrachloro dibenzodioxin, was inadvertently produced. So as part of the chemical process of making these herbicides, a little bit of dioxin was formed and ended up in these formulations. Now, Agent Orange wasn't the only formulation produced. You can see in the barrels here on the lower left, some barrels were painted blue. That would have indicated a different sort of mix of herbicides than Agent Orange. So while we hear about Agent Orange, there were other formulations as well. Here you see an image of the factory. I've forgotten the year this was taken, but it's quite old. So that factory was there doing chemical manufacturing since the 40s. And we all know that, the United States used Agent Orange and other defoliants throughout Vietnam and Southeast Asia during the Vietnam War and perhaps in other places as well. And so those countries have a serious dioxin contamination problem because those herbicides were sprayed widely and on purpose. In Newark, we have the problem because during the manufacture of these herbicides, wastes were discharged into the river. I believe also the facility burned down a couple of times. And so, water running off from that would have carried contamination into the river. And in addition to the dioxin contamination, there are many other manufacturers and industries along the Passaic River, and there are many other contaminants in the river's sediments. But dioxin is one of the most important because of its high toxicity, even at low levels. So these dioxin wastes were reburied because we have natural sedimentation in the river. As I mentioned before, the dioxin sorbed to the sediment particles, so they tend to be transported with the sediment through tidal pumping. And in the 2016, EPA issued a record of decision that the lower 8 miles of the river should be dredged to remove the most highly contaminated sediment and to cap it with clean sediment. Okay, so what my lab has been doing is studying whether these dioxin compounds are transformed by the bacteria in the Passaic River sediments. So dioxins, I've noted them here as PCDDs, but I'm referring to the dioxins. You have them in many different chlorination configurations, right? If they don't have very many chlorines on them, aerobic bacteria that use oxygen can degrade these to CO2. But ones with lots of chlorines on them get dechlorinated, like that trichloroethylene I showed you. So they can be dechlorinated by the organohalide respiring bacteria. That really rolls off the tongue. So here's an example of a couple of different strains of those dechlorinating bacteria and a pathway of chlorine removal from the dioxin molecules. So red is one strain and black is another. And so we see that even closely related strains of dechlorinating bacteria might do different things to these molecules, might knock off different chlorines. So one of the things we want to look at in the Passaic is what kinds of bacteria are there and what kind of pathway of dechlorination is happening. An important thing to note is that if I take this 2378 tetrachlorodioxin molecule and I just take one chlorine off, it is now suddenly not nearly as toxic or dangerous as it was before. And in fact, EPA hardly even measures things like trichlorinated congeners. They're more interested in the ones that have 4 or more chlorines on them. So dechlorination of the dioxins has a very interesting aspect in that it could greatly reduce the toxicity. Or if we started with a molecule with more chlorines, we could inadvertently produce more toxic congeners. So it's very important to understand the fate of these compounds in the river. So we did a lot of microcosm studies, and to try to grow up a lot of dechlorinating bacteria, we fed them other things like trichloroethylene that we talked about before, or dichlorobenzene that you see there on the right. It's just an aromatic compound with 2 chlorines on it. And the Passaic River mud dechlorinated both of those types of compounds. So it dechlorinated trichloroethylene very quickly to ethene. So we know there are a lot of these organisms in the sediment. We then fed our enrichments and the sediments different congeners of dioxins, the tetrachloro, a dichloro, and another model compound down at the bottom that has all the chlorines on one ring. And here's my student, Rachel Dean. She finished her PhD a couple of years ago, and she works down in Washington, D.C. for the EPA right now. But she set up many, many microcosms using that black Passaic River mud that you see. So this is a very busy graph, but just want to take home that what we saw is that if we feed Passaic River sediments some dioxin, the 2378, we do see dechlorination down to some dichlorinated congeners. You can see the timeline here, how patient Rachel was. This was after 884 days, so she certainly deserved to finish her degree and get to leave. Another important thing we noticed is that if we feed sediments a dichlorinated congener, it will take all the chlorines off. And so we greatly, greatly reduce the toxicity of these compounds. So our overall findings show that dechlorinating organisms in the Passaic River do dechlorinate dioxin. but it's pretty slow. And that makes sense because that dioxin contamination has been in the river for almost 7 decades. So our questions now, and we're going on with our study, is how could we harness these dioxin dechlorinating bacteria for bioremediation? How do we stimulate them to go faster? What do we need to add to the sediments to help them do what they're doing, but do it faster? What sorts of things do they need us to add to their environment to be successful in bioremediation? Okay, now I have a few things. I'm not sure if Amy wants me to stop or talk about what we can do at home.
Yeah, well, this was very informative. Thank you. So I do have a couple of questions. So why don't we take a couple of those and then we can definitely dive into what people can do because I know everyone is interested in that. So thank you.
Okay.
So let me get these questions just a second. Okay. So let's see. So Jamie wants to know, are there examples of microorganisms that have been found in contaminated sites that have evolved over the years to be genetically different and possibly more efficient than naturally occurring ones? In other words, do contaminated sites provide their own environment for rapid evolution of microorganisms?
Well, that is such a great question and such a fundamental question, right? So when these dechlorinating organisms were first discovered, many people thought it might be because they had evolved to have this ability relatively recently, because humans had, on the grand scale of the life of the earth, humans are just a little blip. So in that final little blip, we started polluting environments like crazy, and maybe microbes evolved quickly to take advantage of that because they use them to grow, right? But it turns out that there are many, in the case of these dechlorinators, there are many naturally chlorinated, brominated, and iodinated compounds throughout the earth. Sometimes they're made by plants, sometimes they're made by little worms, by sponges. So it turns out that these dechlorinating bacteria have probably been around since the beginning of life on Earth. And because they have evolved to use these many different types of extremely low concentrations of naturally chlorinated compounds, I didn't talk about the genes involved, but they have in their genome, they encode for a dozen or maybe up to 3 dozen different enzymes to attack and remove the chlorines from different types of molecules. So they seem to have a suite of enzymes that they've evolved over time with. And at a site that is contaminated with a much higher level with something like trichloroethylene, maybe the organisms that happen to have an enzyme that can knock the chlorines off of that are selected for. So our pollution selects for, organisms that are better able to do that. So first, they seem to be quite old organisms. The other thing is that they seem to swap these genes. So they are able, it seems that there may be something called lateral gene transfer going on with these organisms. And so they swap out enzymes with each other. Also, of course, engineers and scientists are very, very interested in trying to genetically engineer these organisms. In addition to things like dioxins, we've got now fluorinated compounds as very important and difficult pollutants. Could we genetically alter dechlorinators to become defluorinators? So there's several groups around the world working on that, trying to figure out how to enhance the genetic capability of these organisms.
Okay, thank you. Now, Joe wants to know, do they continuously take samples during remediation at contaminated sites in order to know if what they're doing is working?
Yes, that's a great question. Yes, they do. There is so much data from these sites that you cannot even imagine how, you know, yes, they are continually sampling. I talked about natural recovery, right? Let nature take its course. But people don't want to hear that. We're not going to, they don't want to hear, we're not going to do anything. It's taking care of itself. So natural recovery is usually in the industry, it's called monitored natural recovery, meaning we're going to go out and sample and make sure that the contamination isn't moving towards someone's drinking water well or toward a municipal water supply. So yes, monitoring is done routinely. So you can probably, if you drive around New Jersey, you can see technicians, engineers, and scientists out in trucks in the middle of the field where they're sampling groundwater wells, or they're taking samples of sediment cores out of a river like the Passaic. And then that material has to be shipped to specialized labs to have the chemical analytical work done to see what the pollutants are. So yes, they track the pollutants. They also track things like, is the environment aerobic, or is there sulfate, or is there nitrate? So they try to figure out what is the environmental condition, what's the pH, what nutrients are present. So there are reams and reams and reams of historical data. And part of that site remediation includes that very, very extensive sampling and analysis, and then reporting out of that data to regulators, to clients, and to the public.
Okay, thank you. Why don't we go ahead with the next part, and we'll take more questions at the end. Thank you.
Okay. So Amy asked me to think about bioremediation at home. And so I was trying to think how we use microorganism, or how could we help remediation. But first of all, I just want to say, anytime you go for a walk, go for a hike, you can find microbes doing work. All right. So if you go to a wet, swampy area, this is at Mill Pond near the Earth Center. I could stand at the edge of this water and watch bubbles coming up from the sediment. Well, that's the activity of microorganisms, of bacteria degrading the natural organic matter that's in the pond sediment, leaves and dead vegetation, right? And they're turning that into CO2 and methane, and they're making biogas, and that is bubbling up from that sediment. You could also, take a stick and stir it and really see the bubbles come up. So we can see through this method and others, the microbes working in the natural world everywhere we go. Another obvious thing is we use microbes to make compost, right? So we can compost kitchen scraps, food scraps, to turn it into compost, and that makes use of microorganisms. If we think about biostimulation in that case, composting is an aerobic process, meaning we need to add oxygen to the microbes so they can degrade this organic matter. So we need to stir the compost, or tumble it in a tumbler if you buy one from a special one, or just if you're in a outdoor container or a tub, you just stir the compost, and through that stirring action, you're adding oxygen to that material to biostimulate the microbes. And I was looking for some guidance, and of course through extension, for the New Jersey Agricultural Extension Agent system, there's many helpful documents, but also tomorrow, I guess, there's a better backyard composting seminar in Cape May. So I know that you can learn a lot about that from the folks around an extension around the state. The other important thing is to keep harmful toxic compounds out of the environment in the first place. So we may be using solvents at our home, paint thinners, things to degrease auto parts, Many things that we would handle very carefully in the lab, we may have sitting on a shelf somewhere in our garage or in our basement. So whenever you're ready to get rid of, dispose of something that you know to be, that you wouldn't want to pour out on the ground because it could make its way into soil, it could make its way into groundwater. Many municipalities will have a household hazardous waste drop-off day. So you can save those things and take them to be properly disposed of and keep them out of the regular landfill. You don't put them in your regular dumpster, so they don't go to the landfill where they're released, and then they go into landfill leachate, which might be going to a wastewater treatment plant, and then any remaining pollutants out into a body of water, but rather to try to collect those and dispose of them so they never enter the environment. And then another thing, if I look at I haven't mowed my lawn yet. I keep telling everybody I'm observing no mow May, but actually just haven't had time to mow my lawn with the end of the semester and commencement. But, another thing is when we select lawn amendments, to select those that are biodegradable in the soil. And then we're using bioremediation at home by making sure that anything we put on our lawn is able to be taken care of by the microbes in the soil that could make sure that those compounds get degraded or converted to CO2, and that they don't further run off into stormwater or into a local creek or body of water. And again, there's another great extension site on organic lawn care that you can look into. So those are a few of the examples that I found on how bioremediation relates to us at home. I'd like to end with acknowledging funders that funded me and my students through many, many years of work on this, and then show you some pictures of the students: Rachel Dean, Haider Alminwali, Cassidy Snyder, JC Miranda, and Eric Nitzberg there on his skateboard, and my collaborators. So thanks to the students for doing all that hard 800-day, 300-day, 500-day work to study these microbes.
Hey, Donna, I'm gonna start the poll so that everyone can take that before dropping off. So let me just get that going, but we do have some questions.
Great.
And I do certainly appreciate you recognizing cooperative extension, and Michelle and I are the lead authors on the organic land care.
Oh, that's great.
So thank you for
Shout
bringing
out.
that up. But yeah, please take the poll while we're talking. It should have popped up. Hopefully people will start taking that. But thank you, Donna, for sure. This is great. Are you ready for some more questions, or do you have another slide?
No, I'm finished.
Okay. All right. So Kathy wants to know, is there any concern that if these bioremediation solutions are introduced on a large scale, that polluters or the government will have less incentive to stop polluting or to rein in those polluting activities?
Yeah, I think it's hard to believe that we have this legacy in New Jersey of contamination. Many of the sites that our students go out and work on as consultants, these are old sites, they're decades old. But we do often hear, we're hearing of the perfluorinated compounds, new contaminants that are now, we see are more recent, but they are also widespread contaminants. It's hard to answer that question, but I think the answer is no. The expense and the time involved in site remediation is incredible. Every piece of real estate that changes hands in New Jersey has to undergo an investigation to make sure there's no contamination, because nobody wants to buy that environmental liability. So people who own properties, I don't think they would ever intentionally contaminates something thinking, oh, this is easy to clean up, because none of it is very easy or very short. There are other methods of remediation. Some involve adding really strong chemical oxidants. That has a shorter timeline, but it's still very expensive, time consuming. And then you have all the monitoring that has to be done. So it's hard to say why we see, I think that the train wreck is a good example. They had the wreck, they did something that they thought was a good idea that turned out maybe it was, maybe it wasn't, but accidents happen. Companies may be following regulations but could still have an accident. So that's a great question. I don't think I've done a very good job answering it, but I don't think remediation is considered an easy way out under any circumstances.
Yeah, well, thank you. So I've had a couple questions asking if your slides will be available. Do I have your permission to send out a PDF?
Yes, I think that's fine. Yes.
Okay, well, we can definitely get your up-to-date slides and
Okay. I'll
things.
give you my up-to-date ones.
We can discuss that. And also the recording of this presentation will be posted on our website probably within a week or so. I will put that in the chat so everyone can see that. But I've got some comments coming in. This was an excellent and informative presentation. Thank you so much. Learned a thing or two here. So we really appreciate you coming and speaking plainly about a very complicated topic. I'm sure that was it's difficult to talk about very technical things in a general manner. So thank you.
Yeah, I mean, I try to talk to my son about this all the time, and you guys are much better listeners.
Well, you had a captive audience here. But yeah, everyone is sending you compliments, and we really appreciate you supporting the Earth Day Everyday webinars. And for our viewers, please come back and see us next week. We have the Marine Extension Program seminar series with how your dog could be responsible for closing your favorite beach, as well as some information about the New Jersey Watershed Ambassadors Program. So that's what we have next week. Looking forward to that. So thank you so much for joining us tonight on a very beautiful evening. And certainly check out our website and you can see our past recordings. And there's also some information on our website about how to donate to this program if you're interested, because this is a free program provided by Rutgers Cooperative Extension, but we would love to have some support. So thank you so much for supporting us. And I'm still getting more compliments, so thank you so much.
Thank you.
Everyone seems very happy, and so we really appreciate you coming to speak to us tonight, and hope you recover from graduation and the end of the
Yes.
semester. So thank you very much. Thanks everyone for joining us, and hopefully we'll see you next week.
Thank you.